Hi, everybody. Today we're going to talk about neurons and signaling in the nervous system. Okay. Have you ever participated in a stadium wave? Think about that stadium wave. We're going to take a quick look at a stadium wave as an example in case you're not familiar with what a stadium wave is. So let's take a quick look at this stadium wave. Okay, so again, a stadium wave is where you kind of stand up and you sit down as the wave travels around the stadium. So kind of think of that and keep that in the back of your mind as we think of how an action potential moves down a neuron. So as we talk about neurons and neuronal signaling and how neurons work, think about that stadium wave as you think about the ion channels opening and closing. So this little guy here is a cone snail, and he actually kills his he kills his prey with venom that will disable neurons. So basically blocking that signal transduction pathway. So a cone snail will kill its prey with venom that disables and paralyzes its food, and so it can go ahead and eat and kill it. So it will catch its prey with that venom. So if we look at the nervous system, first of all, we need to have an understanding of neurons in order to understand how the nervous system works. So a neuron is going to be a single nerve cell, and so that nerve cell will transfer information in the body. And they're going to be electrical signals. So they're going to be very, very quick signalings. And so the electrical signal is going to go a long distance down an axon. And it's then going to send a chemical signal from one neuron to another. So it's kind of a combination between two types of signaling that's happening in those nerve cells. So the long distance message is going to be sent with electrical signals, and we'll see how it does that with ions moving across the membrane. And then it will send a chemical message, neurotransmitters, from one neuron to another. When we've got a cluster of neurons together, that's called a ganglia. And then when that ganglia is much more complex and organized, then that's a brain. So our brain is a collection or cluster of nerve cells that all work together. And then when we look at the organization of the nervous system is kind of organized into different types of neurons. We've got sensory neurons that are going to be picking up sensory information from the environment. And that information is then sent to interneurons, which are going to integrate and process that information. So that's usually the brain or the spinal cord where that information is integrated and processed. And then messages go back out to the body through motor neurons, and that's going to trigger either muscle or gland activity. So that's going to cause a response as a result. And then we've got the central nervous system and we've got the peripheral nervous system. So the central nervous system is going to be where that integration and processing takes place. So again, that's your brain and your spinal cord. And then the peripheral nervous system is going to be all of the stuff that comes off of your brain and your spinal cord going out to your body and your extremities. So it's on the periphery or the outside of your central nervous system. And then when we look at those neurons, they're going to be bundled together in the form of nerves. So a nerve is actually several bundle of neurons all working together to send a message from one part of the body to another. So this diagram kind of sums up that whole process, that the design of the nervous system. So we've got sensory neurons picking up information from our senses, our eyes, our ears, our touch. All of our sensory neurons are picking up messages that get sent to our interneurons and the brain and the spinal cord for integration. And then that's going to lead to a response. So it's going to send out a message to our muscles so the muscles can contract and respond. So your central nervous system is going to be your brain and your spinal cord. Your peripheral nervous system is going to be your sensory neurons, and your motor neurons bringing information in and sending information out. In order to understand how the nervous system works, we kind of need to have an understanding of the structure of a neuron. And so the neuron is going to have a cell body that's going to be kind of the large part of it that you're going to find the nucleus and most of the organelles in the cell body. And then there are going to be tentacle-looking things coming out of the cell body, and those are going to be your dendrites. So your highly branched extensions, and that's where you're going to be receiving the signal. So the dendrites in the cell body are receiving the signal, and then the axon is going to be transmitting the signal. So that's usually a lot longer of a little tentacle-looking structure. So a long tentacle coming out of the cell body, and that's going to transmit it to other cells. Where the axon is attached to the cell body is called the axon hillock. So if we look at this diagram here, we can see that the dendrites are going to be at the top here, and so they're going to be picking up the signal. The cell body has the nucleus and other organelles in it, and you've got the axon hillock where the axon is attached to the cell body, and the axon goes a long distance, and the electrical signal is going to go down the axon in one direction and one direction only until it gets to another cell, either another neuron or a muscle cell or a gland cell. And so the two neurons are not going to be actually touching. There's a teeny tiny space between each of those neurons. And so that is called a synapse, the gap or the space between neurons. And then the chemical message is going to be sent across the synapse, and that chemical message is called a neurotransmitter. So a signal is going to go in one direction. It's going to be picked up from the dendrites. It's going to go down the axon, and then it's going to go across the synapse to the next neuron. So because you've got that synapse there, the first cell is called the presynaptic cell. And then after the synapse, you've got your postsynaptic cell, because again, you're going in one direction only. So the synaptic terminal is going to be the end of the axon where the neurotransmitter is secreted from. And so the chemical message is going to be sent across that synapse, that junction between the two axons. So again, the presynaptic cell is the one that comes first, and the postsynaptic cell is the one that comes after it. The postsynaptic cell could be another neuron, it could be a muscle, it could be a gland. When we look at an action potential, a message going down the axon, it is going to be transmitted. It's an electrical signal that's going to be transmitted down the cell. And to understand how that electrical signal is sent, we kind of need to look at the charge inside and outside of the cell. And so that's going to be the resting potential of a neuron. So the difference in charge between the inside and the outside of the cell is called the membrane potential. And when the cell is not doing anything, not sending a signal, it's just kind of hanging out there, it's resting, that is the resting potential. And the normal resting potential of a neuron is going to be negative inside the cell and positive outside the cell. So there's going to be that charge difference between the inside and the outside, and that is the resting potential. So normally when the cell is at rest, when you're at resting potential, what you've got is a high concentration of potassium inside the cell and a high concentration of sodium outside the cell. Both of those are positively charged, but the inside the cell is normally negatively charged, and that is because there are protons and other large molecules inside the cell that are negatively charged, making it negative inside and positive outside. We've got the sodium-potassium pump, which we know already uses ATP, so that's going to be active transport, and that sodium-potassium pump is actively working to pump sodium out and potassium in to the cell. And so we're going to keep the high sodium out, high potassium in, and the sodium-potassium pump is working to keep it that way. That's going to change when an action potential comes, but the resting potential, high potassium in, high sodium outside of the cell. And then when we're looking at action potentials, we've got ion-gated channels opening and closing. So those ion channels are going to be embedded in the cell membrane of the neuron all the way down the axon. And so at resting potential, those ion channels are closed. The sodium-potassium pump is working to keep sodium out and potassium in. And there are negatively charged molecules inside the cell, keeping it negative inside and positive outside. So normally you've got lots of potassium inside the cell, you've got lots of sodium outside the cell, and then you've got large proteins and other molecules inside the cell, making it negative inside and positive outside. So if we look at the cell membrane of the neuron, you've got a whole bunch of different ions all the way across, ion channels. So here's your sodium-potassium pump working and pumping sodium out, potassium in. You've got potassium channels, you've got sodium channels, and they run all the way down the whole cell. So let's take a quick look at a video that kind of sums up what we just said.
A resting neuron, one that is not conducting signals, contains potential energy resulting from an electrical charge difference between the inside and the outside of its plasma membrane. A resting neuron, one that is not conducting signals, contains potential energy resulting from an electrical charge difference between the inside and the outside of its plasma membrane. The cytoplasm just inside the membrane is negative in charge, and the fluid just outside the membrane is positive. The membrane is said to be polarized. Like a flashlight battery, the difference in electrical charge carries potential energy, which can be measured as a voltage. The oscilloscope shows this charge difference, called the resting potential, in millivolts. A neuron has a resting potential of about minus 70 millivolts. What causes the resting potential?
The plasma membrane of the neuron keeps large negatively charged molecules such as proteins inside the cell. Pumps and channels in the membrane regulate the flow of positively charged sodium and potassium ions. The neuron maintains a higher concentration of sodium ions outside the cell than inside, and a higher concentration of potassium ions inside the cell than outside. In a resting neuron, most sodium channels are closed, but many potassium channels are open. As a result, very little sodium can diffuse into the cell, but potassium ions can flow freely out of the cell, adding to the positive charge outside. The sodium-potassium pump actively transports more sodium ions out of the cell than potassium ions into the cell. This contributes to the buildup of positive charge outside the cell, while keeping the concentration of sodium low and the concentration of potassium high inside the cell. The diffusion of positive potassium ions out of the cell and the large, negatively charged molecules trapped inside the cell cause the charge difference that constitutes the resting potential.
Okay, so we've got that resting potential, negative inside, positive outside. So when we've got an action potential going down an axon, that is going to be an electrical signal going all the way down, and that's going to be caused by changes in that membrane potential. And that's going to be a response to a stimuli. And so when you have a stimuli that causes an increase in that membrane potential, and it makes it more negative inside, then that's going to be a hyperpolarization. If you've got a signal that is going to make it more positive inside, that's going to be a depolarization of the cell membrane. And so with hyperpolarizations, you're looking at even more negative inside. That is not going to send an action potential. But if you've got a depolarization, making it more positive inside the cell, that is what is going to initiate an action potential, but only if it reaches a certain threshold. So if a signal is not strong enough, if it doesn't send enough of those positive ions into the cell, it's not going to send a signal. But if it does bring enough positive ions into the cell to make it positive enough, it reaches a certain threshold, then an action potential will be triggered, and then that is going to initiate a wave down the axon, so kind of like a stadium wave down the axon that is going to send a signal onward. So that would then initiate an action potential. So again, hyperpolarization, you're making it even more negative. That is not going to send an action potential. But the depolarization, you are increasing the charge on the inside, making it more positive. And if it reaches a certain threshold, then it will initiate an action potential. And so we're looking at ion channels opening and closing, and like a wave down an axon, that is going to cause an action potential. So an action potential is that signal being sent down the axon. So if we look at that action potential, again, triggered by that depolarization that reaches that threshold, we can take a look at how an action potential works. So we're starting off at that resting potential. And so if we look at this diagram here, here's your resting potential. So negative inside, positive outside, the ion channels are closed. So both the sodium and potassium ion channels are closed. Then when an action potential is generated, so a signal is received, that is going to initiate the voltage ion channels to open. So the sodium channels will open. And when the sodium channels open, the sodium ions are going to rush in. So they're going from high concentration to low concentration across the sodium ion channels. So the channels open, and that's going to cause it to become more positive inside the cell. And what's going to then happen is once that ion channel has opened and the sodium has rushed in, that's going to trigger the next neighboring sodium ion channel to also open, allowing even more sodium ion channels to open. That's going to cause an action potential because we're now above the threshold that's becoming positive on the inside of the cell. And so that's going to cause the charge inside to be positive. Once an ion channel has opened and the sodium ions rush in, it's going to close shortly after. It's going to cause the next one to open and then close. And so we're going to have those ion channels opening and closing like a wave all the way down the axon. Right behind that wave, we're going to have another wave where now the potassium ion channels open and the potassium ions leave the cell. And so that's going to bring the charge back down to negative inside because all those potassium ions, positive ions, are leaving the cell. And so that's the falling phase. So it's dropping back down to negative inside the cell. And oftentimes, you'll have more of the potassium leaving than sodium entered, so you get this kind of overshoot. So that little purple part is the overshooting of the ions. But then it's going to kind of level off because what's happening is the sodium-potassium pump is now pumping the sodium back out and the potassium back in. So you've got these waves. The sodium ion channels are opening, triggering the next one to open, and then they're closing. So it's opening, ions are flowing in, and then it's closing, which is triggering the next one to open and close, which triggering the next one to open and close, like a wave all the way down the axon. So you've got a wave of sodium channels opening and closing, and then right behind it, you've got a second wave of the potassium ions opening and closing, and the ions are flowing back out. So first the sodium ions flow in, and then the potassium ions flow out. So it becomes positive inside the cell, and then it becomes negative inside the cell again. So it starts negative, goes positive, and then goes back to negative. And so that is an action potential and how an action potential works. And so if you're looking at the membrane potential, the charge inside, it's going to be negative, and then it goes positive when an action potential goes down, and then it goes back down to negative. So you've got the sodium channels opening and then the potassium channels opening, and then it goes back to resting potential. But now the sodium is inside and the potassium is outside. We need to reset it. And so the sodium potassium pump is what's going to be doing that resetting. It's going to pump the sodium back out and the potassium back in. And so that's kind of why an action potential can only go in one direction, because there's a refractory period. So after an action potential goes, you can't initiate a second action potential because the sodium channels are temporarily inactivated. And the sodium is now in the wrong place. It has to get reset. So the sodium has to go back out. The potassium has to go back in so that it can send another signal. Another signal is not going to come because you can't, even if the sodium channels were to open, there's no more sodium to flood in because it's already gone in. So it has to be reset before another action potential can go. And so that's why it only travels in one direction. So let's take a quick look at this animation that kind of sums up that whole process.
We've looked at the resting potential in a neuron that is not transmitting nerve signals. What happens when the cell is stimulated? A stimulus may change the permeability of the cell membrane to sodium or potassium ions. This alters the distribution of charge in the cell body, and if the change is great enough, it triggers a signal called an action potential that moves down the axon. How does the action potential move along the axon? Voltage-gated channels in the membrane open and close in response to voltage changes across the membrane. When no action potentials are being transmitted, most of these channels are closed. An action potential begins when voltage-gated sodium channels open and sodium ions rush into the cell. The plasma membrane becomes positive on the inside and negative on the outside. Very quickly, the sodium channels close, while voltage-gated potassium channels open, allowing potassium ions to rapidly diffuse out. The plasma membrane returns to being positive on the outside and negative on the inside, and the potassium channels close. Meanwhile, the sodium ions inside the cell have diffused to adjacent areas, causing a slight change in the polarity of the membrane ahead of the action potential. This change in polarity causes the voltage-gated sodium channels along this part of the membrane to open. Again, sodium ions rush in, and the action potential spreads to the adjacent part of the neuron. In this way, the action potential travels down the neuron like a wave. The efflux of potassium ions during the falling phase of the action potential restores the negative charge inside the neuron. Meanwhile, the sodium-potassium pump has been shuttling sodium ions out and potassium ions in, re-establishing the resting distribution of sodium and potassium ions.
Let's look at another action potential as it moves along the membrane. This time we will measure the voltage changes that occur in one spot along the membrane. A slight change in polarity causes the voltage-gated sodium channels to open. Sodium ions rush in, causing a reversal in membrane polarity. Sodium channels close and potassium channels open. As potassium ions rush out, the membrane returns to being negative on the inside. The sodium-potassium pump then restores the balance of ions present at resting potential.
Okay. So again, an action potential can only travel in one direction because you've got the sodium channels opening, sodium rushing in, and then the potassium rushing out. And so that has to be reset. So the sodium potassium pump is going to reset it. Plus you've got the sodium channels are going to be temporarily inactivated. So you can only have the action potential working in one direction. So one direction only. So this diagram kind of shows that how the flow, the wave down the axon, and so you've got your action potential initiating the sodium channels opening and then closing, and then the next zone they're going to open and close, and right behind it, excuse me, you've got the potassium channels opening and closing and opening and closing. So just like a wave down an axon, you've got the sodium channels opening and closing, then right behind it, the potassium channels opening and closing. So sodium rushes in, potassium rushes out. If we look at how neurons are sent in invertebrates, we have some of our neurons are going to be coated by a myelin sheath. And so there are Schwann cells that will produce the myelin sheath. That's actually going to increase the action potential speed. And how it does that is it kind of puts this protective coating, but you've got spaces or gaps throughout the neuron. And so the action potential will actually jump from one space or gap to the next. And so it doesn't have to open and close as many ion channels to send that wave down the axon. And so it can send a much faster signal if it has that myelin sheath. So some of our axons are covered in that myelin sheath to increase the speed at which an action potential can move down an axon. So much faster with the myelin sheath than without it. This picture is just kind of, again, summing up that wave as the action potential moves down the axon. Okay, when the signal reaches the end of the axon, then it needs to send a chemical message across the synapse to communicate with the cell that's next to it. And so the electrical signal is coming down the axon, and when it gets to the end, it is going to cause calcium channels to open up, which is then going to cause the synaptic vesicles that are containing the neurotransmitters. They are going to merge with the cell membrane. So in exocytosis, it's going to release the neurotransmitters, which will then diffuse across the synaptic cleft, which will then get picked up by the receptor proteins on the postsynaptic cell, and then it's going to trigger possibly another action potential in the next cell if the signal reaches that threshold potential. So this diagram here is kind of showing you that synapse. So you can see the axon at the top here, and the signal, the action potential, will come down the axon. And when it gets to the end, it's going to cause or trigger these calcium ion channels to open. calcium will flood into the cell, which is going to trigger the synaptic vesicles to move and merge with the cell membrane, spilling out the contents of the vesicle, which is the neurotransmitters. They diffuse across the synapse, and then they're going to attach onto ligand-gated ion channels that are on the other side. So the neurotransmitters are your ligands, and they're going to cause those ion channels to open, which is going to trigger a response in the next cell, the postsynaptic cell. So again, the neurotransmitters cause the ligand-gated ion channels in the postsynaptic cell to open, which will then cause a response in that next cell. So different responses can happen. It could be a stimulating response, it could be an inhibitory response, but some kind of response will happen as the neurotransmitters bind to the next cell. So an excitatory postsynaptic potential is going to be depolarizations that bring it towards the threshold. And so if the threshold is met, then it's going to cause an action potential in the postsynaptic cell. But some signals are going to be inhibitory postsynaptic potentials, and so they're going to cause hyperpolarizations or move it farther away from the threshold, making it more negative inside the cell, and that's going to prevent an action potential from starting in the next cell. Those neurotransmitters that are released, they don't stay there forever. So they kind of need to go away so that another signal can come later. So what will happen to the neurotransmitters? One of three possibilities. It could be that they simply diffuse away, so they drift off and they leave the synaptic cleft, or they could be taken up by the surrounding cells. So the cell that released it could pull it back in and then recycle and reuse them. Or there are digestive enzymes in the synapse that will then break down and destroy the neurotransmitter, so they're not constantly being attached onto the postsynaptic cell and causing a signal to be received. So this diagram kind of sums that up. So here you can see the reuptake or the reabsorbing of the neurotransmitters. Or here you've got digestive enzymes that are breaking down the neurotransmitters, or they simply just diffuse away and drift out of the synapse. So those are typically the three options that will happen to a neurotransmitter. Okay. So most neurons are going to have many synapses on their dendrites and their cell bodies. So you're usually picking up a bunch of signals. So usually one signal is not enough to trigger an action potential. So usually it's an accumulation of multiple signals that are being received on the cell. Okay. So here you can see several different signaling neurons that are being picked up by that particular neuron here. And so it's going to be an accumulation of them. So they have to reach that threshold in order for an action potential to actually go by. There are lots of different neurotransmitters. So there's more than 100 different neurotransmitters. They usually fall into different groups. So it could be acetylcholine, it could be a biogenic amine, it could be an amino acid, a neuropeptide, gases. All of those are different types of neurotransmitters. So lots of different neurotransmitters, lots of different receptors for those neurotransmitters. Acetylcholine is going to be a very common neurotransmitter. So in research, you're going to come across acetylcholine a whole lot. Acetylcholine is involved in muscle stimulation, memory formation, and learning. So it's a very common neurotransmitter that you'll see often in research. So lots of different neurotransmitters. And here's just a diagram kind of summing up that neurotransmitter system. And so you've got your your calcium channels opening, which is triggering the merging of the synaptic vesicle, the neurotransmitter spilling out, and then being picked up by the receptor on the other side, leading to a cellular response. Then again, the enzymes are either degraded or reabsorbed or drifting away. Okay. So here's a quick animation just to sum up the synapse. So let's take a look at that.
Neurons communicate across junctions called synapses. When an action potential arrives at the end of an axon, a signal is transmitted across a synapse to a receiving cell. Although synapses may be either chemical or electrical, we will only look at a chemical synapse in detail. How do signals travel across a chemical synapse?
There is a narrow gap, the synaptic cleft, between the synaptic terminal of a sending neuron and the surface of a receiving cell. An action potential arrives at the end of the axon of the sending cell. This causes chemical changes that make vesicles containing a chemical called a neurotransmitter fuse with the plasma membrane. The neurotransmitter molecules spill out into the synaptic cleft, diffuse across, and bind to the receptors on the receiving cell's membrane. The binding of neurotransmitters to receptors causes the attached ion channels to open. Depending on the type of neurotransmitter, receptors, and channels involved, this may excite or inhibit the receiving cell. Here we show excitation. Sodium ions rush in, changing the polarity of the membrane, and an action potential is triggered in the receiving cell. The neurotransmitter is broken down by an enzyme, and the ion channels close. Enzymatic breakdown is one of the several mechanisms by which transmission is terminated at chemical synapses.
And here's a quick animation that kind of sums up neurons. Let's take a look at this one.
This baseball player springs into action, his nervous system rapidly processing information in a network of nerve cells, or neurons. Each neuron receives input from one or more cells. In response, the neuron may generate an electrical signal known as an action potential that travels down the length of the axon. Let's take a closer look at the plasma membrane of an axon. Even without an action potential, the axon is a busy place with many ions moving across its membrane. Much of this ion movement is driven by the sodium-potassium pump. Using energy from ATP, sodium-potassium pumps actively transport sodium ions out of the cell and potassium ions in, creating an uneven distribution of charge across the membrane. Some potassium channels are open all the time, allowing potassium ions to leave the cell. As a result of these ion movements, the inside of the cell is negative relative to the outside. This condition is called the resting potential. The membrane of an axon is also packed with gated ion channels that open and close during an action potential. At resting potential, the gated channels are closed. If a stimulus changes the distribution of charge across the membrane sufficiently, the gated sodium channels open. Movement of sodium ions across the membrane makes the inside of the cell more positive. This reversal of the charge distribution causes the gated sodium channels to close and the gated potassium channels to open. As potassium ions move out of the cell, the original charge difference is re-established across the membrane, closing the gated potassium channels. This sequence of events is called an action potential. The sodium-potassium pump restores the distribution of ions back to their previous levels at resting potential. Let's look at how the action potential is conducted along the axon. As the change in charge difference across the membrane spreads from open sodium channels, other sodium channels farther along the axon begin to open. The original sodium channels close and adjacent potassium channels open. As potassium ions move out of the cell, the original charge difference across the membrane is restored, and then the potassium channels close. Meanwhile, new sodium channels open, followed by the opening of new potassium channels and the closing of sodium channels. In this manner, the action potential is propagated along the axon of the neuron, eventually reaching another cell. The information carried by this action potential will be processed with other information, permitting this baseball player to make a spectacular catch.
Let's take a quick look at this next one. But that's a nice animation kind of showing you how that action potential goes down the neurons. Let's take a look at this last one.
Our brains are constantly processing information, which requires communication between billions of nerve cells. Signals are passed from a sending neuron to a receiving neuron at a junction called a synapse. An action potential in the sending neuron travels down the axon until it reaches a synaptic terminal. The narrow gap between the synaptic terminal and the receiving neuron is called the synaptic cleft. The synaptic terminal of a sending neuron contains numerous vesicles filled with neurotransmitters, chemicals that carry information across the synaptic cleft. When an action potential reaches the synaptic terminal, the vesicles fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters affect the receiving neuron, changing the distribution of charge across its membrane. Let's take a closer look. An action potential is propagated down an axon by the opening and closing of sodium and potassium channels. When an action potential arrives at the synaptic terminal, it causes the opening of calcium channels, shown in green. Calcium ions enter the synaptic terminal through the calcium channels. Calcium ions bind to the vesicles containing neurotransmitters. This causes the vesicles to fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters diffuse across the synaptic cleft and bind to receptors in the plasma membrane of the receiving neuron. Here, the receptors are ion channels that open. Ions move across the membrane, changing the distribution of charge across the membrane. The neurotransmitters are quickly removed from the synaptic cleft, ending their effect on the receiving neuron. A single neuron can receive signals from many sending neurons. The signals arriving from the axon on the left are excitatory. They make the receiving neuron more likely to generate an action potential, as indicated by the green glows. Signals from the axon on the right are inhibitory. They make the receiving neuron less likely to generate an action potential, as indicated by the red glows. The two sets of signals cancel each other out, and no action potential is generated. Now, only excitatory signals are transmitted, and the receiving neuron generates an action potential down its axon. The signals that pass between these and countless other neurons constitute our thoughts and coordinate our activities.
Okay, so nice animation to kind of sum up both how an action potential works and then how a synapse works. And you're seeing inhibitory and stimulating signals being sent. And so it does take a lot of axons to actually initiate another signal being received by that postsynaptic cell. Okay, so that kind of sums up the action potential, and it kind of ties both signal transduction pathways in with our membrane potential and diffusion across the membrane. So it ties our last unit in with this unit, and a nice way of seeing how signal transduction pathways work. I hope this was helpful, and I'll see you next time.